Hi everyone and welcome to the internet. So today's lesson is going to be a little bit different because it's going to actually be a bit of a double lesson. So what we've got is mutations first of all, and then we're going to go on to have a look at the history of genetics towards the end. To start off though, we've got 3 questions based around our previous lesson, so answer those in your book for me. Okay, let's mark those then. Question 1, the dominant are shown by a capital letter, mark for that one. For question 2, both alleles are the recessive alleles, tick for that one. And then for number 3, if you got the gametes for the mother as the 2 X's in individual circles, 1 tick. If you got the father's gametes as 1 X and 1 Y, tick for that one. If you got the 4 genotypes, so XX, XX, XY and XY, tick for that one. If you got the 4 phenotypes, so 2 female as the XXs and the 2 males as the XYs, tick for that one. And then if you wrote down the percentage as 50% chance of male or 50% chance of female, give yourself a tick for that one. So give yourself then a mark out of 7 and write that in your books. First thing I'd like you to do then is write down what you think a mutation is. When we talk about mutations in biology, what we're referring to is a change in the base sequence in the DNA. So if you got that, give it a tick. If you didn't get that phrasing, make sure you've written that down as the textbook definition, if you like, for it. So a change in the base sequence in the DNA. See if you can write me down a list of anything that you think causes mutations. Just 1 minute to have a go. When it comes to mutations, obviously you can mark any of these you got right and make sure you've got these written down if you didn't have them. The first and most important point we need to remember is that mutations can occur spontaneously. So throughout your life, then there will be mutations that occur within your DNA. But we also see that there are certain factors that increase the chance of a mutation occurring. And the two key ones that we really need to remember here, firstly, ionizing radiation, and a really good example of that is UV, which comes from the sun. And the second one are exposure to certain chemicals. So things like ethanol, things like benzene, these will all increase the chance of mutation occurring. Have a go answering this then. Why would sunbathing increase your risk of skin cancer? Hopefully we managed to put together the idea that if you're sunbathing, then you're increasing the exposure of your skin to that ultraviolet radiation, the ionizing radiation. Now, what happens is when your skin cells are exposed to that ultraviolet radiation, then it can lead to damage in the DNA of your skin cells. And that damage can lead to skin cancer. And that's why it's so important that when you are outside, then you are wearing sun cream of an appropriate sun protection factor. Because anytime you're actually burning yourself, then what you're doing is that's your body's way of saying you've caused damage. It's a bit late to do anything about it, hence why we use the prevention of the sun creams. So whenever you're going out in the really nice weather we've had of late, then sun cream is important. What we've discussed in some of our earlier lessons on genetics is this idea of variation. So we've talked about the fact that we do have variation within a species. Now, the reason that we've got that variation is because we've got a different version of an allele. And that's arisen from a mutation that changed the DNA. So where we've got all of these different eye colours, skin colours, hair colours, everything like that, they've all come from mutations within those particular alleles, leading to these different phenotypes that we now see. But not all mutations will show the same effects. When we get a mutation, some of them will show absolutely no effect on the phenotype at all. So what we see is even if you have a mutation, it won't always present as a change in the phenotype, the characteristic we can see. It may influence the phenotype, so it might be a contributing factor, but not the overwhelming deciding factor, if you like. So it might just give a little bit of a change. Or it may overall determine the phenotype. And it's that last group there, where the mutation determines the phenotype, that we get those variations within a species. So make sure you've got those points written down. I'll leave it up for another minute and a half. Now, as I've mentioned, throughout your life there will be a mutation or two that occur within your body. But it's not a case of if you have a mutation then it's instant death. Because mutations come into three categories. We've got harmful mutations, we've got beneficial mutations, and we've got neutral mutations. And we're going to look at each of those in turn just to see what we mean by those and some examples of them. If we start off with the harmful mutations first of all, which are probably the ones that your brain jumps to straight away, then the first really good example of a harmful mutation are the ones that cause cancer. And where we talk about cancer, this is basically a condition in your body where your own body cells are growing and dividing in an uncontrolled manner, which is why it's so tricky to treat, because any of the drugs that we're using that target the cancer cells, well, they're just normal body cells. That's why we've got so many side effects with cancer treatments there. Second harmful mutation are the ones that cause abnormal channel protein formation. Now, channel proteins, if we think back to our earlier biology work, are these kind of little channels that go through the cell membrane. So they're a specific shape to allow certain molecules to cross from one side to the other. And if that channel protein isn't produced normally, so we make a different shape or we don't make enough of them, for example, then we get problems in that process. And a good example of a condition that is caused by this abnormal channel protein production is cystic fibrosis. The third one there is that we can actually cause different shape protein molecules to be made. And a good example of this is sickle cell anemia. So your red blood cells are normally that biconcave disc shape if we've not got sickle cell. But if you do have sickle cell anemia, they change from that disc shape into more of a crescent moon shape, so shaped like a sickle. And that means that they're not going to be as efficient at transporting oxygen around your body. So make sure you've got those examples written in your book. I'll leave them up for about another 40 seconds. When we talk about neutral mutations, these are ones that have absolutely no bearing on your chance of existing and surviving at all. So good examples of this is your ability to roll your tongue and whether you've got 3 earlobes or whether they're attached to the side of your head. It really makes no difference whether you can roll your tongue or not. It's not a case of if you can roll your tongue, you're definitely going to survive to the age of 90, whereas those who can't roll their tongue, they're doomed by the age of 60. It's just something that we can either do or can't do, but it doesn't impact on survival. The third type of mutation we've got are those that are beneficial. Now, if we consider one example that's not in humans first of all, so something that occurs in bacteria, we've got that mutation for antibiotic resistance. So that means that bacteria now won't be killed by the antibiotics that we take to get rid of them. So it's beneficial to the bacteria, not so much for us. Second one is the FOXP2 gene, and this was a mutation that allowed us to be able to speak. So if we hadn't had the mutation in the FOXP2 gene, we would not be speaking as we do today. And the third one on there is the skin colour mutation. So hopefully we know that if we track back our human history, then we originate from a small number of breeding pairs that originated on the continent of Africa. Now, if you are in Africa, it's actually beneficial to have much darker skin because that protects you from absorbing too much ultraviolet from the sun and therefore reduces your risks of skin cancer. When we moved to lovely climates like the UK and obviously those places away from the equator, then the amount of ultraviolet in the sun was decreased. So the intensity of the sun's rays as we move further from the equator does decrease. And actually having darker skin isn't an advantage there. So we had this mutation within the genes that control skin colour that led to the lighter skin tones, and that means that when we're living in countries like England where for some years it's pretty much cloudy and rainy almost all the time, then we can still absorb enough of the sun's rays to make enough vitamin D to stop us getting conditions like rickets, for example. So mutations in skin colour are beneficial as well because it's allowed us to live successfully in a range of different countries around the world. Have a go at answering the question on the screen in your books then. Let's double check to make sure we got the key points. So hopefully we know that people with darker skin then have reduced amount of UV absorbed by the skin cells. And it's more important that this is true of the skin cells deeper within the actual body, because those are the ones that are more susceptible to damage. Because they're absorbing less of that UV with those delicate cells, then they're less susceptible to damage, which means you reduce the risk of skin cancer. If we have a mutation to give lighter skin, then in those countries that have lower intensities of sunlight, then the skin can still absorb enough to make vitamin D, and people with the darker skin tones in countries that are quite dark and dingy, then they can actually struggle to absorb enough to make the right amount of vitamin D. So some people may actually have to take supplements of vitamin D to actually stave off those conditions that require vitamin D to avoid. If we consider these mutations in a bit more detail now, what we've actually got, if we look at a strand of DNA, is it's organised into two different sections. We've got coding sections, so they're the bits that actually code for specific proteins, they're the genes, and they're separated by these non-coding sections. So what we can see is we've got different regions along the DNA strand, we've got the genes, the coding sections, separated by the non-coding sections. When we're talking about a mutation, we know it is a change in the base sequence of the DNA. But there are three key ways that this can occur. The first one is that we can change an individual base. Second, we could add a base into the sequence. Or third, we could delete a base from the sequence. So jot those points down for me about what the mutations could actually be. Then we'll look at each one in a little bit more detail. Before we leap into what's actually going to happen, then see if we can remember the process by which we make a protein, so protein synthesis. See if you can explain how a protein is made. So you get your first mark if you said the DNA for the gene unzips. Second mark, 3 RNA bases in the nucleus pair with the complementary base pairs on the DNA. Third mark for saying the mRNA strand leaves the nucleus and travels to a ribosome. Fourth mark, the ribosome reads it in triplet codons or sequences of 3 bases. Next mark is for saying that each triplet corresponds to a tRNA molecule which is attached to an amino acid. Next mark is for saying the tRNA brings the amino acid in and they join together to make a peptide chain. And the final mark for saying that it then folds to make the protein. Give yourself a mark out of 7 based on that, and obviously if you want to write this down as a reminder, hit pause on the video and just jot those points down. What we're going to do then is have a little look at each of those in turn to see what impact it actually has. So at the top I've given you our original DNA strand, and the first one we're going to look at is changing a base. So what we can see on the second strand, the one that we've had the mutation occur in, then the T that we had as our second base has now become a G. So that's our mutation, that one single base being changed. Now because we've changed that T to a G, what we've done is we've changed that first triplet, because it no longer reads ATA, it now reads AGA. And that means it's not going to code for the same amino acid, necessarily. Amino acids have more than one triplet code, but it can be that we've changed it to a completely different amino acid here. So what we see there is we've modified that one triplet, which could mean that we've changed the amino acid it's coded for. So that one amino acid will change, which could have obviously significant impacts on the protein. So that's our first one. Second type of mutation is where we add a base. So we've got the same initial DNA code there, but this time after the T we've added another T. Now the problem of this is that we've not only changed that first triplet codon from ATA to ATT, but it's changed the other ones as well, because everything's been shifted along one section. So what we see is we've, rather than having ATA, then GCA, then TGC, etc., we've got ATT, AGC, ATG, and so on. So it's not just changed the first triplet, but it's changed all of the triplet sequences that follow it. So this is going to have much wider reaching impacts than just a single substitution of a base. Third one is where we delete a base. So again, we're looking at that T. So we've deleted the T, and that again means we've not only changed that first triplet, but we've changed all of the triplet sequences that follow it. So either adding or deleting a base changes every triplet codon past that point in the sequence, whereas our substitution of a single base just changes an individual codon. The big issue with changing these triplets and therefore changing the amino acids we code for is that when we change amino acids, it can impact on the way that the protein actually folds up. So it changes the three-dimensional picture or structure of the protein. And that means we could actually create a completely different protein. And that's obviously problematic because proteins can be things like enzymes, and the shape is very important to those. So let's see if you can tell me why then. Have a go at answering this question in your books. If a mutation occurs in an enzyme, then as we've said, the three-dimensional structure of the protein is changed. Now that means that the shape of the active site could change, and that means it's no longer complementary to the substrate. So the substrate can't bind to the active site because they're different shapes to one another. The enzyme can't work. As we mentioned earlier, we have these non-coding sections between each of our genes. And in that non-coding section is a really important triplet called the start codon. So it's a sequence of the 3 bases that literally tells the cell this is where you start to make the protein from. Now, if we have a mutation that occurs within one of those start codons, it's actually going to potentially change it so it no longer says start here. If we don't have that, it's kind of like having an ignition without the key. It's not going to actually begin anything. So what we see is if we have a mutation in the start codon, then the gene may not be transcribed at all, which means the protein will not be made at all. So this means that we just wouldn't make a particular protein as a result of that mutation. Before we move on to the history of genetics, then let's just double check our knowledge of these 3 points. So 3 questions for us to answer based around our first part of the lesson. Part 2 of our lesson then, the history of genetics. First thing I'd like to do is just jot down the names of any scientists you know who've been involved in the development of our knowledge on DNA. As we go through these, obviously you can tick off any that you got right. We're going to start all the way back at the beginning, if you like, of our DNA story. And to do this, we're going back to 1866. So perhaps not as far in the past as you may have thought. And we're actually going to go and visit Gregor Mendel. Now, Gregor Mendel was actually someone who did lots of experiments with pea plants. And what he did was he observed that characteristics were passed on from the parent pea plant to the offspring pea plant. So by observing these characteristics in his experiments with his peas, then he noticed, first of all, that characteristics in plants were determined by hereditary units, so units that are passed on from one generation to the next. Those units were passed on one from each parent, so both of the parent plants were involved in passing this information on, and those units were either dominant or recessive. Now it's because of this work that we actually refer to Gregor Mendel as the father of genetics. He was the one that spent years, as you can imagine, pea plants don't grow overly quickly, crossing different peas with one another to see the outcomes and documenting it all to work this out. So summarise those points in your book. I'll leave them up for about another 45 seconds to give you a chance. Scientist number 2 is Friedrich Miescher, and in 1869 he discovered a substance called nuclein. Now this was an acidic substance that he discovered in the nucleus, and he then named it nuclein. So he was the one that found there was something acidic within the nucleus. The third scientist to join our genetics party is Oswald Avery, and his work was carried out in 1944, so we had a bit of a gap between our last ones. And he actually was looking at bacteria, and he worked out that you can transfer DNA between different bacteria, and that then meant that the ability to cause disease was passed from one strain of bacteria to another. And then those bacteria passed on the ability to their offspring. So from that work, he worked out that genes are made of DNA. Six years later, in 1950, we've got Erwin Chargaff. Now, he discovered base pairs. He did a lot of work looking at DNA in different organisms and found that even though different organisms have different amounts of DNA, that no matter which organism he looked at, it all had equal quantities of adenine and thymine, and then equal amounts of cytosine and guanine. So even though the overall amount of DNA may have been different, the proportion of adenine to thymine was always the same, and the cytosine to guanine always the same. So see if you can answer the question at the bottom when you've summarised. Hopefully what we remembered is that those are the complementary base pairs. So if we've got adenine at 20%, because that always pairs to thymine, you must have 20% thymine. So because they're complementary to one another, we have the same amounts of those base pairs. Two years later, we had two scientists, Rosalind Franklin and Maurice Wilkins, and they were doing a lot of work on actually taking photographs of DNA crystals. And the way they did this was by using X-rays. And the very important picture you can see is the one at the middle of your screen there, which is the one that went on to then help the next advance. But Rosalind Franklin was the one that did most of the work on this, and she produced that image that we see there. And that tells us about the structure of the DNA itself. Now, unfortunately, because she spent a lot of her time working with X-rays, which it's not great to expose yourself to in great quantities, then she actually developed cancer early and did die before she really got recognised for her contribution. Without the work that Rosalind Franklin had done on getting that image of the DNA, then our next two scientists, James Watson and Francis Crick, couldn't have come up with their DNA double helix structure that we know. Because what they did was they used her image to then come up with this structure that we know today as our double helix. So the two spirals held together by the complementary base pairs. And from their work, they won a Nobel Prize. But sadly, because obviously Rosalind Franklin had died prior to that, she was never able to receive recognition in that form. Between 1953 and the year 2000, we've carried on looking at DNA. So we've been identifying individual genes that code for inherited disorders like cystic fibrosis. And we've also developed a whole new field of science called genetic engineering. And we're going to look at genetic engineering in a lot more detail in B6. So it's not just been a case of we discovered the structure of DNA and we were done and just left that on a shelf. We've carried on looking to see what more we can discover about it. One of the significant things that's occurred in the more recent past was in 2003, when we completed the Human Genome Project. Now, this was a rather ambitious project at the time, which was over 20 years. So not just something that's done in a short space of time, 20 years of work went into this to sequence all 24,000 genes of the human body. So the goal of this project was that we're going to sequence every gene in the human body, and they did it. That was completed in 2003. Once we actually knew where all these different genes were in the human genome, then we could start to work on something called gene therapy. So that's where we can identify which genes in certain conditions are faulty, so they're different to the normal gene if you like, and we can develop techniques to allow us to replace those faulty genes with normal copies and therefore treat certain genetic conditions. Last thing I'd like you to do then is just to sum up what we've done in terms of the history of genetics as a simple timeline. So draw a line down the side of your page, and then at the top you can start with the earliest date and then work your way down with the key dates, the scientists, and the key summary of their discoveries. So I'd suggest you take a little bit of time to do this, but obviously I'm not going to leave this on the screen forever. I'll leave it up for another minute, but take your time, work through it and create a good timeline, because that means it's a very nice quick check to make sure you've got the right dates, scientists, and what they contributed.